Hi, I'm Dave Champlin. I'm a professor of biology at the University of Southern Maine, and this video is about transcription regulation of gene expression, and in particular, during cell differentiation. The two pieces of paper represent two cells from any species of eukaryote. They could be from a human or any other animal, or these two cells could be from any plant. Or they could be from any single-celled eukaryote, like the yeast that causes bread to rise, for example. The specific example I'm going to use are two human cells. One is a human muscle cell, the other is a muscle precursor cell called a myoblast. In the first month of pregnancy in humans, there are no muscles. We each inherited from our parents the DNA information to form muscles in our entire body, but we're not using that information to form muscles yet. It's in the second month of pregnancy in humans that muscles start to form. These particular images are of a mouse embryo that's a strain that has genetic information that's been tagged so that when muscle cells form, the cells glow red. The muscles glow red. In this strain, the tendons glow green, and so forth. In the early embryo, the cells are not glowing. In general, early embryos have undifferentiated cells and then undergo a process of differentiation to form differentiated cells like muscles, skin, kidney, brain, etc. Our focus on this is going to be in the DNA in the cells, and the DNA is going to be represented by this clay. The blue clay represents the familiar double-stranded DNA helix. The blue clay would typically be thousands of nucleotide letters long, the xxxxxxx letters. The DNA is floppy like that, as it is in our model. DNA is floppy and more like cooked spaghetti than uncooked spaghetti, and you'll see that's relevant in our story. There are two reasons I'm using clay. The first reason is because this story is very physical. Although DNA is too small to see clearly even with the most powerful electron microscope, nevertheless DNA is physical. Stories about DNA are always physical stories. My research includes a lot of work with DNA, and I think I like my work partly because I grew up in a small farming town in Minnesota where everything was very physical. Tractors would break and people would fix them. Crops would grow and they'd get harvested. DNA has that same physical logic. I feel like I'm a car mechanic who works on physical DNA. I often tell my students that if a car doesn't start, nobody gets out and looks at their tires. DNA has that same physical logic. Here's a quick example. Scientists can take a test tube and they can actually purify the DNA away from any cells and have purified DNA in that test tube. DNA is physical and it is able to be physically isolated from all the other molecules of life. In the test tube, scientists can study this physical DNA. They can identify physical mutations that have occurred to the DNA. They can read the DNA sequence, the pattern of xxxxxx letters. They can compare that sequence between two people or between any species and work with this physical DNA. A pretty dramatic example is that in a test tube, scientists can physically cut DNA in specific locations, and then they can mix and match DNA between two species. In every species on Earth, DNA has the same chemical structure and the same nucleotide letters xxxxx. The difference in the DNA between different species is in the order of the nucleotide letters along the DNA strand. In this example, I glued onto the DNA this green strand of DNA, which is actually from a jellyfish. And this DNA can be placed back in cells and studied in that context. Here's an example that's pretty common of this story. In just about any pet store you go into now, you can find some fish that are called glowfish that have had this jellyfish gene incorporated into their own chromosomes. This fish inherited that gene from its parents, and it will send it on to any offspring. The jellyfish gene is called GFP, and when it gets expressed into proteins, the protein glows. In fact, this mouse strain is glowing because there have been several different versions of the GFP gene incorporated into the mouse genome. The second reason that I'm using clay in this work is because research in how students learn has shown that in science, students learn science topics well by building models out of paper, paint, colored pencils, clay, and labeling those to explain the physical concepts found in science. So that's why we're working with this, and we'll show how this is important during differentiation. The most important concept is shown here already, and that is during cell differentiation, in general, the DNA doesn't change. So we've got DNA that looks the same in both the myoblast and the muscle. Even though we're going to zoom in on a gene called the myogenin gene in our example that's critical for muscle differentiation, the myogenin gene, and none of the DNA changes during differentiation. This story is general. In general, the DNA doesn't change in cells. So for example, the DNA is the same in a muscle cell, a brain cell, a liver cell, and pretty much every cell in our body has the same DNA. In fact, it has all the DNA needed to make the whole organism. That concept is called genomic equivalence, and what it means is that in each cell in our body, we have one entire genome. In fact, in humans, we have two genomes. A genome is the amount of information necessary to make a particular species, and we inherited one human genome from our moms and one human genome from our dads. Each of our cells has genomic equivalence. That's true in every plant and animal. So in a plant leaf cell and a plant root cell, for example, they have the same DNA, even though the cells are very different. That's true also in examples like this, where when cells are treated with medicine, the DNA doesn't change. Medicine can have a dramatic effect on a cell, but one thing it doesn't affect is the DNA. In general, we think that we're born with the DNA that we'll have throughout our whole lives. There are some exceptions to that rule. Here's a really important one. In comparing a normal and a cancerous cell, mutations have occurred to cause this cell to become cancerous. It's true that during our lifetime, we will acquire a small number of rare and random mutations, but in general, we think that we have the same DNA as a baby, as a teen, and as an adult throughout our lives. The DNA doesn't change, and like I said, the myogenin gene doesn't change, and that's true for genes in general. During cell differentiation, the DNA doesn't change, and the genes don't change. In the second video, I'll explain what does change for myogenin during differentiation. But for now, we're going to close this out by labeling these parts of the myogenin gene. The yellow, I should switch back from the jellyfish to the mouse myogenin gene. The yellow is the transcription unit. That's part of DNA. The myogenin gene encodes a protein, so the transcription unit has a coding region. There is a region of the DNA that's far from the transcription unit that's called the enhancer, and there's a region right next to it that's called a promoter. None of these change because the DNA doesn't change during differentiation, and these terms are all referring to parts of the DNA, parts of the gene. We'll deal with what does change in this story in the next video.